Immunology Insights, Basics and Beyond, Chapter 44, CAR T-Cell Therapy. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our previous chapter, we explored the concepts of immunoediting and immune evasion, how the immune system and cancer are locked in a constant battle, with tumors finding clever ways to escape immune control. Today, we shift from the natural immune response to one of the most revolutionary weapons in modern oncology, CAR T-cell therapy. This is one of the clearest examples of using engineering to supercharge the immune system, turning a patient's own T-cells into targeted cancer killers. It's science fiction brought to life, and it's already saving lives. CAR T-cell therapy not only represents a medical breakthrough, but also shows what's possible when gene therapy, cell biology, and clinical oncology work hand in hand. So let's talk science. What is CAR T-cell therapy? Well, CAR T-cell therapy is a form of adoptive cell transfer in which a patient's or donor's T-cells are genetically modified to express a chimeric antigen receptor or CAR that specifically recognizes a tumor antigen. A CAR is made of three main parts. First, the extracellular targeting domain, usually derived from an antibody fragment, scFv, that binds a specific antigen on the tumor cell surface. Second, the transmembrane domain, anchoring the receptor in the T-cell membrane. And third, the intracellular signaling domain, typically CD3 zeta, plus co-stimulatory domains like CD28 or 41BB, which activate the T-cell after binding, bypassing the need for MHC presentation. And this is a key advantage. CAR T-cells can recognize tumor cells even if they have lost MHC class I expression, which is a common immune evasion strategy. How does the therapy work? Well, let's look at this step by step. First, we have the leukapheresis. T-cells are collected from the patient's blood in a process similar to dialysis. In autologous therapy, these are the patient's own T-cells. In allogeneic or off-the-shelf therapy, the T-cells are taken from healthy donors, engineered to avoid graft versus host reactions, and stored ready for use, drastically reducing wait times and costs. Second comes the genetic engineering. Most current products use viral vectors, so lentivirus or retrovirus, to insert the CAR gene into the T-cells. Alternatives include non-viral methods, such as sleeping beauty transposons that use a synthetic transposase enzyme to cut and paste the CAR gene into the T-cell's genome. It's relatively inexpensive and scalable. Or piggyback transposons, which follow a similar principle, but allow larger genetic payloads and may integrate more efficiently. Both require delivery into T-cells, often by electroporation, which on the other hand can be toxic as the electrical pulse and the high DNA amounts may cause significant cell death. And then we have CRISPR-Cas9, which can precisely add the CAR gene to the T-cell's genome or knock out undesired genes, for example PD-1 to boost the function or TCR to make cells universal. Third, we have the ex vivo expansion. The modified T-cells are activated using, for example, antibodies against CD3, CD28, artificial APCs, or other expansion reagents, and grown in cytokine-rich media, often containing IL-2, IL-7, or IL-15 or some combination, to drive proliferation and improve survival after infusion. This expansion step can take 1 to 3 weeks in autologous products. Fourth, we have the conditioning chemotherapy. Before infusion, patients receive lymphodepleting chemotherapy, usually fludarabine and cyclophosphamide, to reduce immune competition and make space for the incoming CAR T cells. And fifth, we have the infusion and attack. CAR T cells are infused in high numbers back into the patient, where they then circulate and locate and destroy tumor cells expressing the target antigen. On recognition, they release then perforin, granzymes, and cytokines, initiating a targeted immune assault on the cancer. What are the clinical applications of CAR T cells? Well, CAR T cell therapy has been most successful in hematologic malignancies. Examples include: first, B cell acute lymphoblastic leukemia or BALL with Kymriah targeting CD19. Second, diffuse large B cell lymphoma DLBCL with Yescarta also targeting CD19. Third, mantle cell lymphoma with Tecartus. Fourth, multiple myeloma with Abecma and Carvykti, both targeting BCMA. Response rates are remarkable. In relapse refractory BALL, complete remission rates can exceed 80%. However, relapse is still possible and therapy is not effective for all patients. Eligibility is typically limited to patients who have failed multiple prior treatments and are strong enough to tolerate potential side effects. Costs are substantial. List prices often exceed $370,000 for the cell product alone, excluding hospitalization and supportive care. Beyond cancer, CAR T cells are being, for example, trialed also for HIV, targeting conserved viral envelope proteins to eliminate infected cells, or hepatitis B, aiming at surface antigens to clear infected hepatocytes, or acute viral infections in immunocompromised patients, for example, adenovirus or cytomegalovirus, where rapid viral control is critical. In these cases, CAR T cells work by redirecting cytotoxic activity towards infected cells, similar to tumor killing, but the challenge lies in avoiding damage to healthy tissue. Let's look at the challenges and limitations of CAR T cell therapy. First, we have the cytokine release syndrome, or CRS, driven by IL-6 and other cytokines released during massive CAR T cell activation. It can cause fever, hypotension, and hypoxia, treated with tocilizumab and corticosteroids. Second, we have neurotoxicity, or ICANS, confusion, seizures, and cerebral edema, likely linked to endothelial activation and blood-brain barrier disruption. Third, antigen escape, loss or mutation of the target antigen, for example CD19 loss in B-cell malignancies. Fourth, solid tumor barriers. Physical and immunologic obstacles can reduce CAR T-cell infiltration and efficacy. Fifth, manufacturing delays. Autologous products require weeks, during which aggressive disease may progress. 6. Logistics and costs. Centralized production is slow and expensive. Point-of-care manufacturing could help. Let's have a closer look at allogeneic versus autologous CAR T-cell approaches. In the autologous approach, the patient's own T-cells are used. They have a low rejection risk, but low manufacturing time and may fail if the patient's T-cells are of poor quality. In the allogeneic approach, donor-derived T-cells are used. They are gene-edited to remove the native TCR to avoid graft versus host disease and the MHC molecules to evade host rejection. These edits, often done with CRISPR-Cas9, create so-called stealth CAR T cells that are invisible to both donor and host immune systems. The field is actually moving towards universal off-the-shelf CAR T cells with rapid delivery, potentially at lower costs, enabling earlier intervention before disease becomes unmanageable. The field constantly evolves and there are some interesting next-generation approaches. First, dual-target CARs. They recognize two antigens to reduce antigen escape. Second, so-called armored CAR T cells. They secrete cytokines like IL-12 to counteract tumor suppression. Third, CAR natural killer cells or CAR NK cells. They are safer, faster to produce, and naturally allogeneic. Fourth, non-viral gene delivery. Sleeping Beauty and Piggyback enable cost-effective, scalable manufacturing, and CRISPR-Cas9 adds precision. Fifth, on-site manufacturing, which could cut production from weeks to days, increasing access and survival chances. Let's assess further the future directions. CAR T-cell therapy is a paradigm shift in oncology, programmable immune cells with curative potential in otherwise terminal diseases. It can be combined with checkpoint inhibitors to improve persistence, or even with chemotherapy to reduce tumor burden before infusion. In the future, we may see, first, a broader use in solid tumors, improved trafficking, local cytokine release, and engineered resistance to tumor microenvironment suppressions are the key here. Second, application to infectious diseases. For example, targeting persistent viruses and intracellular pathogens with antigen-specific killing. Third, in autoimmunity, selectively removing pathogenic immune cells by targeting their unique receptors. Fourth, in transplant tolerance, eliminating only the immune cells responsible for rejection. And fifth, with alternative cell types, for example, gamma delta T cells, natural killer cells, and the similar sounding but different NKT cells may offer better tissue penetration and lower graft versus host disease risks. To conclude, CAR T-cell therapy shows the incredible potential of merging biotechnology with immunology. By equipping immune cells with synthetic antibody-like precision, we can bypass tumor evasion strategies and deliver a targeted strike. The field is rapidly evolving towards faster, safer, and more universal products, bringing this once last resort treatment closer to frontline care. In our next chapter, we will explore immune checkpoint inhibitors, drugs that release the brakes from the immune system, letting T cells do their job with renewed force. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time and thank you for listening.